How do molecular TB tests find genomic mutations? Imagine trying to find tiny spelling mistakes in a massive book. That's similar to how molecular tuberculosis tests identify genetic mutations in the bacteria that cause TB. These tests analyze the DNA of Mycobacterium tuberculosis to spot specific changes linked to resistance to drugs. The process starts with collecting a sample, often sputum from a patient, which contains the bacteria. From this sample, scientists extract the DNA, which is like copying the bacteria's genetic blueprint. Next, they use a technique called polymerase chain reaction, or PCR, to make millions of copies of specific parts of the DNA. These parts are carefully chosen because they are known regions where mutations often happen if the bacteria become resistant to drugs. For example, mutations in the RPOB gene are linked to resistance to rifampicin, a key TB drug. Similarly, changes in the CATG and INHA genes are associated with resistance to isoniazid. By amplifying these regions, scientists can focus on the most important parts of the bacteria's genome. Once the DNA is amplified, the test looks for mutations using different methods. One common approach is line probe assays, which use special probes that bind to either the normal or mutated sequences. If the bacteria have a mutation, the probe will bind differently, revealing the resistance. Another method is DNA sequencing, which reads the exact order of the nucleotides, the building blocks of DNA. Sanger sequencing can identify known mutations, while next-generation sequencing provides a more detailed picture, even detecting mixed bacterial populations that might include both resistant and susceptible bacteria. Whole genome sequencing takes this a step further by reading the entire bacterial genome. This comprehensive approach can find all kinds of genetic variations, including single nucleotide changes, insertions, and deletions. It helps scientists understand how the bacteria are evolving and spreading. Although whole genome sequencing requires more time and resources, it offers the highest level of detail and accuracy for detecting drug resistance. After identifying mutations, the results are interpreted based on established scientific knowledge. Some mutations are well known to cause high-level resistance, while others may have a more variable impact. These molecular results are faster than traditional culture-based tests, often providing answers within a day or two, which is crucial for starting effective treatment quickly. In summary, molecular TB tests find genomic mutations by extracting bacterial DNA, amplifying key gene regions with PCR, and then detecting mutations through hybridization or sequencing. These advanced methods allow for rapid and precise detection of drug resistance, helping doctors choose the best treatment plan. Always remember, for any diagnosis or treatment decisions, consulting healthcare professionals is essential to ensure the most accurate and personalized care.